baby.
Hey, when we're done, you know how early we watched that other video about DNA, replication, all that stuff, the video thing?
Yeah.
Can I show you one that's like the same guys and they're singing the Backstreet Boys when we're done?
Okay.
So
DNA's replication. Are we watching a video again?
No.
Let's see. This is. We're talking about DNA a little bit here, which you should already know because we talked about it when we did organic molecules.
Wait, are we doing that right now? The DNA replication? So
Yeah.
wait, what's that?
So if you look, chapter 13, page 227. 227. This is the chapter 13 essays. Tell me about DNA.
Chapter 13 essay.
13.
Do you have this one?
What's up, Troy?
Yeah.
Here.
I think I might have this one.
The acetyl-CoA attaches to a C4 molecule.
I would.
Acetyl-CoA is a 2.
See, look.
It'll attach to the C4 and now form a C6.
Oh, there's one.
Alright.
Yeah. Those are pretty cool.
That's kind of what I'm saying now.
I'm looking at this later. I'm just gonna
Yeah, I think I put too much information in there.
Yeah, you put way too much.
Okay. We know through numerous experiments, we're on page 227. We know through numerous experiments which they went over in chapter 13.1. The DNA is made up of
What chapter is this? I'm sorry.
13, page 227. DNA is made up of these thingies. Thingamajiggers. Thingy doodles.
Nucleotides.
Nucleotides. Now, the one I'm holding is just looks like this, but in actuality, if you could see the molecule up close, it would look more like this. These are nucleotides. A nucleotide consists of 3 parts. A sugar, which is called deoxyribose. That's where DNA gets the name. D comes from deoxyribose. That's the name of this sugar. The 5-carbon sugar. They actually number the carbons. Do you remember that? 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. 5-carbon sugar. No,
I remember.
So it consists of a sugar. That's a phosphate group. If you remember our study of functional groups, chapter 2. And there's a nitrogen base at the top. Now the nitrogen bases actually come in 4 types. These ones with the double ring structure are called purines. Adenine and guanine are the purines bases. And they have double ring structures. And how do you remember that? People from Georgia are pure.
People from Connecticut are pyros.
No.
And these are the pyrimidines, which, as you can see, still have the sugar and the phosphate, but only a single ring nitrogen base. People from Connecticut are pyromaniacs. Pyrimidines is what we call these. That's how I remember those. Now, whenever you see these in a molecule, they connect together according to certain rules. A purine always bonds to a pyrimidine. And they actually do it specifically. A, adenine always bonds to thymine, and G, guanine always bonds to cytosine. A to T, G to C.
Alright, Georgia Tech.
No.
No. Grilled cheese, GC.
Yeah, never mind.
Georgia Tech would make things a whole lot more confusing and wrong.
Yeah.
Yes.
Yeah. Georgia
Yeah,
College.
Georgia College.
Georgia College.
Growing carrots. Angry.
Grilled
Just
cheese.
remember at, and then there's
Angry
GC.
tomato.
Yeah.
Tomato.
Remember at, and
Grow
then the other
carrots.
ones are together.
Coach, there you go. Angry tomatoes grow carrots.
Okay. Whatever works. Now, so they connect together like that, A's to T's and C's to G's, and they form, the bonds that they form there in the middle are weak hydrogen bonds. Pretty weak bonds there in the middle.
Weak sauce.
And so what it does is it makes it easy for the molecule to tear apart. And so this thing will unzip like a zipper. Now the bonds that connect one base to the one below it are very strong bonds. They're really hard to break.
What? Say that again. Sorry.
The ones that connect.
Okay.
The bonds that connect right there, see where I'm pointing?
Yeah.
Those are real strong. Those are covalent bonds. Whereas the ones that connect this way are weak hydrogen bonds. Covalent bonds are a lot stronger than hydrogen bonds. So if you heat this molecule up, get it vibrating real fast, it will tend to tear apart like that.
Where do they connect? Like on the nucleic acid, like in the picture. Like what's attached to each other.
That hydrogen and that hydrogen connect to fall off in an OH. I guess you have to put, I'm not sure. They connect base to base somehow.
Okay. Sorry.
Maybe it's these oxygens here. Hydrogen bond to,
Yeah, that makes sense.
yeah, because they're double bonded. Form hydrogen bonds to these hydrogens, possibly.
That makes sense.
To these hydrogens. I don't know. It doesn't matter. Anyway, so you get the structure now. This thing goes straight up and down, but it also twists. Every 10 bases that you count downward, it does a full twist.
Double helix.
So we call that the double helix.
Every 10 is that?
Every 10 bases, it gives you a full twist.
That's cool.
So this is what it looks like when it's all twisted up. And the guys who discovered that,
Won a Nobel Prize.
you want to remember their names?
Well, no.
Watson and Crick. Very good.
Nice.
Wow.
Okay. There it shows how they
There
bond
it
there.
is.
Those are the three hydrogen bonds between G and
Go
C.
kids.
There's Watson and Crick.
It looked like they didn't name Watson Crick.
DNA staircases, DNA art. Now, the second part of this essay, you need to talk about how DNA copies itself. That process is called DNA replication.
Like the song, we saw that.
Right.
That one.
And how does it do that? Well, you have an enzyme called DNA helicase that comes in and it unwinds the DNA and unzips it. That was my hand, was DNA helicase there.
Don't you usually have one?
There's DNA
I don't know
replication.
where it is. So helicase unwinds, and then another enzyme called DNA polymerase. So there's two enzymes involved here. Helicase does the unwinding, and then polymerase comes in and takes these free nucleotides and adds them on one by one. Click.
Polymerase adds them on
Yeah. Polymerase adds new nucleotides on. Click.
from 5 to 3.
Good point. It can only add them on in the 5' to 3' direction. Now, what in the world does that mean?
It's a good question.
Well, if you look back at the bases, they have, the carbons are numbered. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. The 5 prime end is the carbon that's nearest the phosphate. The 3 prime end is the carbon that's on the opposite side of the sugar. So, if I'm holding a nucleotide like this, there's the phosphate on the bottom. The 5 prime end of this thing is that phosphate end.
Okay.
The 3 prime end is up here at the top.
Wait, so 5 prime equal
The
bottom.
Phosphate, yeah. Well, you say bottom, but remember, these things can be turned upside down. So there is really no bottom. So the phosphate end is the 5 prime end. It's easy to remember, 5 prime phosphate, they have
What does
the
that
F
mean
sound.
though? Like what is
And you're used to hearing the F sound every time you get a test back.
No, I'm not.
If
I
you
got
got 77 on my last test, just want to throw that out there.
92.
Okay, so when I say that these things have to be added on in the 5 prime to 3 prime direction, what I'm saying is they have to be added on like this. Click, click, click. Do you see the 5 prime is on this end?
Uh-huh.
And the 3 prime is on that end?
Uh-huh.
So I have to go that way.
Oh, yeah. Alright.
That's all that means. Oops. You have to write about this if you get this essay. If you leave this part out,
Can we
you're
say
gonna
it?
miss
Like
points.
noise?
Click.
Like
Say the new strand has to be added, has to be replicated in the 5 prime to 3 prime direction.
So, I have a quick question. If we
Yeah.
say what you say, like in the review, will we get a 10 out of 10?
Yeah,
Okay.
probably. Unless I leave something out.
That's not fair.
Like, you almost about 5 to 3 prime. Thank you, very good.
So anyway, on the opposite side, this is the tricky part. It doesn't start at the top
It starts.
and go downward. Like you would think, like it did on this side, it just starts at the top and goes downward. But on this side, it has to start at the bottom,
That's
like right here,
not the way.
and go upward. Because only that way are you going from 5
3.
prime to 3 prime.
And those are called key.
Click,
You had one right there.
click, click. I'm out.
No, that's it. I see him.
Where? It goes with this one?
Oh, maybe not. No.
Yeah,
Okay, we're gonna pretend.
Those are called okazaki fragments.
Yeah, that's an Okazaki fragment,
Whoa.
and as
I
you
just looked in the book.
listen, as this thing unzips even more, you'll make another Okazaki fragment a little bit further down.
But this is the primase that attaches to the.
And what puts each of those nucleic acids on that?
There's an enzyme called DNA polymerase
But what about the primase?
that attaches.
It's a big yellow thing.
You don't have to worry about the primase. It's another enzyme that actually. There's another step that I left out. There's an RNA primer that's added,
Oh,
and then it's immediately removed. So it's.
we don't need to worry about it?
Nah,
No.
you're good.
Alright, good.
What about the
Yeah, I forgot.
the DNA ligase?
Okay, let me get to
sorry,
that.
sorry, okay.
So, here we have, this is called an Okazaki fragment, and this is called an Okazaki
You're like
fragment.
super
So one side is done continuously,
It's a lagging.
and the other side is done in fragments. The side that's done continuously is called the leading strand, and the side that's done in fragments is called the lagging strand.
I like that. That is straightforward and
Yeah.
logical.
And
What's the lagging strand
it's
called?
the side that's done in Okazaki fragments.
The one that's not, it's going slow. It's in
Okay.
fragments.
So,
So it's lagging. And the one that's going faster is
Yeah.
leading. It's in the lead.
That's right.
Makes sense.
I'm like.
Okazaki.
Wait, how many does it do in a thing?
They could call it DNA fragments.
Like they're done in fragments. But is
How
there
many
certain-
fragments?
Well, like how many are in each fragment? Is
I
there
don't
like
know.
a certain number?
What's
I don't know.
Okazaki?
It's an
Okazaki
exact
was the
number.
guy that discovered that this is how it
Right,
works.
was he
Japanese?
Yeah.
Yes, he was.
Huh, knew it.
Really? Wow.
Now,
I thought he was Jewish.
listen, there's one more thing. There's another enzyme that has to come in and glue together the Okazaki fragments. It's a special enzyme that creates this bond. There's a bond, let's pretend this one fits here, I know it doesn't. There's a bond that has to go, a covalent bond that has to be produced right there. And it takes a special enzyme called DNA ligase.
The glue.
So DNA ligase glues together the Okazaki fragments.
Okay.
2 grams of butter.
Once you do this to the entire DNA strand, you have copied your DNA exactly.
I have a quick question. You know how in the book it says that DNA polymerase can't start the synthesis of the DNA chain? Do we have to say how it is actually a strip of RNA?
No.
Okay.
No, you leave that out.
So I read that in my other essay. It's really fun.
Everything gets more technical than I've explained it. Not just this, everything we talk about here.
I think I
Especially
wrote
the
all the
cellular
technical
stuff.
stuff in my essay.
There's a lot of little. You can take a whole course in this. Each day that I lecture, you can take a whole course in.
Really? That's like intense.
What about today?
Okay, so that's how DNA copies itself. That's replication of DNA.
And that is essay.
That's essay. And, well, you talk about the structure of DNA and then the replication of DNA. That's an essay. That was chapter 13.
I can
That
do
is what
okay.
I'm afraid.
Yeah, I feel like this is enough
I can
information.
do it.
Be an essay.
Whatever said before,
Like it's not even a full
me
page.
and Harrison can give you all the.
It's not enough information?
Yeah,
I guess
Oh,
if I
wow.
expand on it
Yeah.
a lot,
you're right on the history.
I just write down like.
I'm not gonna write all the history. I might write. I'll write guidelines for all the history essays.
Oh, yeah.
I wrote so much of my essay.
Or a thesis and like groupings and stuff.
Yeah, I just,
Okay.
yeah, that's legit.
Well, if you need more to write, just look at 13.2. You can write it in 13.3. You can write anything in
What
there.
about
Right.
the you could like mention the is it just like eukaryotic that we're supposed to or like what is
It's just
it?
eukaryotic, but if you need
Words,
words, you can write about prokaryotic.
No, they're just like they're gonna be iffy now. Like where we're
Okay.
You'll see.
Oh.
They're usually like this.
Now this shows that this is kind of neat. This shows that the replication of DNA isn't just happening in one spot. Here we're building some new DNA here.
Oh yeah.
And we're also building some new DNA down here. And in a big long chromosome, it's actually happening in thousands of spots. Still, it takes 8 hours to copy the DNA in a human cell. Even though it's happening in lots and lots of locations. We call each of these a replication bubble, wherever the DNA is copying itself. And you can see it just goes on, and eventually they meet each other. So you can talk about that if you wanted to.
Okay.
How fast does it go? 500 to 5,000 base pairs per minute.
500.
500 to 5,000 base pairs per
That's how
minute.
fast what goes?
That's pretty
That's
big
how-
range. It's pretty big range.
That's how fast this DNA replication goes.
I thought you said it took 8 hours.
It does.
There's billions.
Because there's so many billion, there's 6 billion base pairs to do.
Equal
If
5.
you're only doing 5,000 a minute, it'll take you a long time to do 6 billion.
But you're about to throw out a
But
number.
it says,
in a minute.
it has been calculated that the average human chromosome contains on the average about 140. Oh, okay, never mind.
141.
Million base pairs.
The average chromosome.
Pretty straightforward.
There's 46 chromosomes.
Alright.
Oh, okay, I see what you're saying.
Yeah.
A little more than this would be good.
That's what I do on tests.
Okay?
You can actually keep it.
I think that you need this. Great trip.
So like, this is going
That
on,
was a good trade.
That was
on
good.
like, all the other stuff going on, like.
This goes on in the S phase of interphase. Why would we copy our DNA at all? Why not just not copy it?
Because
We
then
have
you
to
can
copy
make more
it to
cells.
make
You make
more cells.
You have to copy it when the cells want
if
to
your
reproduce.
DNA lists
That's how
cells,
cells
that'd be
divide, quickly.
A human cell that's dividing quickly divides once every 24 hours or so.
Wow.
not like nerve cells.
Nerve cells
Never.
just stop after a while.
And this, you know,
Is that why when
most how
like,
it is, that's like.
Alzheimer's.
or like when stuff like,
Cycle, alright.
never mind.
Muscle cells don't.
I was wondering
Not much, no.
if your skin cells are dead and like, if you like cut yourself,
Uh-huh.
how do they just become dead cells if they need to like?
They're dead on the outside, but down
Well,
deep they're alive.
Like the top ones are dead?
Yeah, the top ones are dead and the deep ones are living.
So you cut yourself.
I mean, I have another really, really random question. I was just watching House and everything, but how do they do bone marrow transplants if the bone is hard?
They get through it somehow.
Big,
A sharp needle
big needle.
or a drill or something.
Oh yeah, bone marrow is not.
Boreal?
Oh wait, no, that's the other one.
That's one that hurts really,
That's why
really
it hurts.
bad.
That's when it hurt. That's one that like.
I'm not sure how they get in there. That's a good question.
Power tools.
We need to ask a
Yeah,
surgeon.
because he,
How can we ask your dad?
He doesn't do bone marrow transplants, but he would probably know.
Yeah.
You want me to call him up?
Yeah.
They couldn't put him to sleep because it was too like taking too long.
Yeah, all the fucking cash cow working over here.
But what?
Nice.
Yeah, I love the upstairs show.
I'm actually
What
do you
I'm huge on cash.
Calling Dad to ask him about bone marrow transplants.
Cool.
I should probably just use something.
No.
It's like, oh my gosh.
Hey Janie, it's Tommy.
How it's made at the same time.
Hey, not a whole lot. Is Dad there? Okay.
This is so funny. Put on speaker.
Tommy, what's up?
Hey Dad. Hey, I'm running a little study session here at Frederica with the kids, and they were asking me a question that I couldn't answer. I thought you might know the answer.
Hey, Dr. Willis.
When they're doing bone marrow transplants, how do they get into the middle of the bone to get the bone marrow?
So disappointed.
It's like uh-huh. He's concentrating.
There's a needle you can pop through the bone into the marrow space, and then you can suck it out. That's what I told them. That's what I was guessing. And now, it must be a pretty hefty needle. It is.
It was the size of this pen on house.
Yeah, it's.
We got people
That's just the needle part though.
They take it from the sternum or the hip.
Ugh, that's so bad.
That's this part. But they take it, they always do it.
They did it from the guy's
They
arm
do,
house.
Like the bag.
They always do it for like the
Uh-huh.
sleep bags. That's not.
That's like that's the hip.
Okay.
They do
Not
like
that.
up here. But that's not.
Well, I guess it is considered.
House is not
Because
because
your
of
hip
that.
is like
House is
right.
awesome.
Is that?
Okay.
To see like.
Yeah. And they also do that with like, you know how they do that and they like.
It was awesome.
That's the.
Oh, okay.
Oh yeah.
You like
Cool.
that?
That's
Yeah.
one of my favorite
You
things.
know how to do that?
I can't watch that forever. Oh, I've watched
That's
that.
Lost.
Yeah, that's just.
Lost. Keep going.
I
She
mean,
was
sure.
kind of loopy
I
afterwards.
never got into it.
So really, when the needle,
And I know that it's like.
Okay, gotcha. Thanks.
Oh my god, no.
Okay. Talk to you later.
My favorite show.
All right, bye. Okay. They give them chemotherapy or radiation, and that kills all their own marrow.
Mm.
And then they stick the needle in and inject the new marrow in. Because you can't get rid of the old marrow. You just gotta kill it.
They did that on House, and they were this lady. She had to kill all of her. They had to kill all of her old.
How do you know you get fired on your day off?
Like in this 4-inch thick room, and it was pretty cool.
To give her radiation so the
Yeah.
radiation won't escape. Okay, whoops.
I went a little bit too far.
Now, so the next part, listen. This is how the DNA code
I like this.
is used to make a protein. This is a protein.
Yeah.
So just to go through the bubble thing, like,
The ribosome.
yeah.
Yeah.
Okay.
So we gotta make this somehow. Now, what's a protein made of?
Amino acids.
A string of amino acids joined by what kind of bonds?
Peptide.
Peptide bonds. Remember that?
Yep.
So how are we gonna make this? I'll put this over here so you can see it. And we gotta make something that looks sort of like that.
Okay.
Okay, so here's the DNA. Now this is
Use an mRNA.
the nuclear membrane right here. This is inside the nucleus, and this is in the cytoplasm.
You can.
And this is a ribosome. You remember ribosomes are all studded along the endoplasmic reticulum?
The rough endoplasm.
The rough endoplasmic reticulum, there's ribosomes all over it.
Yeah.
Little dots.
Uh-huh.
Y'all remember that?
That's
Yeah.
that little dot.
This is one of the little dots.
Okay.
Let's make that with a hole.
Now the ribosome is going to work together with a couple other types of what we call RNA to get this protein made. And I'll show you that as we go along. Now, so how this starts is there's an enzyme, a special enzyme called RNA polymerase. It's like DNA polymerase, but it's RNA polymerase.
Little brown.
DNA polymerase makes new DNA. RNA polymerase makes new RNA. So
Which makes
polymerase, polymerase is that word polymerase has
Many
polymer in it. So polymerase is an ACE means enzyme. It's an enzyme that makes a polymer. So you have DNA polymerase that makes the DNA enzyme, and you have RNA polymerase that makes the RNA.
So it's transcription and translation are the two parts of this protein?
Yes, it happens in two parts. The first part is called transcription, and the second part is called translation.
Take the whole thing. That's just long false secret class. Right.
Okay?
Okay.
So let's do transcription first. RNA polymerase comes in. It opens the DNA up. A lot of kids screw that up right there. They say helicase comes in and opens the DNA up.
But it's polymerase.
It's not. It's RNA polymerase in this case.
It's a 2-for-1 special.
There is no helicase. And
Opens up what?
opens up the DNA.
It's 2-for.
Do we need to know about the promoters?
You can mention it, yeah. Yeah, you can mention it. The promoter is the site where the RNA polymerase attaches. It's a part of the DNA where the RNA polymerase attaches. It's called the promoter. Pretty good there.
Yep.
You got that in your notes?
Yeah, and it also defines the start of the gene, direction of the transcription, and
Yeah.
strand
Don't
that
hold that ball.
is to be transcribed.
Harrison's all over it.
Yeah.
All over like a fat kid on a piece of cake.
So,
Yeah.
RNA polymerase attaches at the promoter, opens up the DNA, and then RNA nucleotides are added one by one. Click, click.
Bloop. Does it have to be 5-3 with RNA also?
Yes, it goes in the 5' to 3' direction.
But doesn't it only copy one string? Or
Exactly.
It only copies one side. That's all we need.
Which is why it mutates more
is
often.
this,
Which is why AIDS is so dangerous.
is
Which
this
is also
the,
in a later chapter.
is this RNA, what attaches it?
And,
RNA?
RNA polymerase.
Does both?
It unzips and attaches.
And these
Now-
are still nucleotides?
These are nucleotides, but they're RNA nucleotides. What's the difference between an RNA nucleotide and a DNA nucleotide?
One's got an R.
One has an R, one has- It's
What's the difference between RNA and DNA?
Yeah,
Not
what's
much.
that
D.
The R and the D.
The ribonucleotide is
Well, yeah.
it's a not D-ribose.
It's not deoxyribose
It's
sugar.
Yeah.
It's ribose sugar. Get rid of the deoxy. That's the difference between RNA and DNA. That's one difference. The sugar is different.
T becomes U.
That's the other big difference.
Oh.
It has U instead of T. Uracil instead of thymine.
Uracil sounds like
How do you spell uracil?
medicine.
Right here.
Does it not?
RNA has uracil instead of thymine. So, take a look up here. If this says on the DNA is TCAAG, then what is it on the RNA? What's added to a T?
U.
A.
Oh.
A and T go together.
It's A.
What's added to a C?
G.
G.
U.
What's added to an A?
U.
Normally you would say T, but RNA doesn't have T, it has U instead.
Another U.
So another U here
C.
and a C here.
Gold. Because that's the atomic symbol for gold.
AGUUC will add to TCAAG.
Gold.
Is that the
Gold
mRNA
carrots.
strand?
This is going to become what we call mRNA.
Ulysses.
What this is going to detach after it's made, and it will then leave the nucleus.
So what
And
would
that is
What
called
detaches, yeah.
mRNA.
what detaches it?
Why? Nothing, it just detaches on its own.
So the copy detaches itself.
So this mRNA will detach, and it will start to leave the nucleus.
Doesn't it do something before it leaves the
Yes!
It adds a tail and a
Base
cap.
and tail.
There has to be some processing going on with this
exons.
mRNA before it can leave.
Exons is enzymes.
There's three major things that happen. We add a cap to one side of it,
So
a
wait,
cap to the 5' end, and a tail to the 3' end.
I never understood the 5' and 3' before now.
How can you make the cap like that?
Like a cap, like
And
A
didn't they
baseball
say
cap.
the
it's
cap?
going this way, it's like a cap.
And don't they say that
Cap's
the tail determines how long it'll live inside of it?
That's a hypothesis. We don't know that for sure.
And the cap is how it enters the ribosome, or
Yeah, it has something to do with the way it either gets out of the nucleus or attaches to this ribosome.
Which end is the cap attached to? The
The
5?
cap goes on the 5' and the tail goes on the 3'.
Is it mRNA when it leaves the nucleus? Is that what it turns into?
Yes, it's mRNA.
so what is it before it leaves the nucleus? Wasn't it something else?
It's free nucleotides.
Okay.
Just chilling.
And where do these free nucleotides chill? Do you know?
In the nucleosome.
In this big round thing in the nucleus
The nucleosome?
called the nucleolus.
Nucleolus, that's what it
Do you
was.
remember the nucleolus?
Yeah, I always thought it was like the nucleus of the nucleus.
Let's go back and look at the nucleolus.
I don't like Nucleolus. I
Wait,
never know
what
how to
is this?
spell it.
What's the
I like the word nucleosome.
Computers can do that?
of the nucleus.
But what is?
It holds the nucleus.
Yeah, baby, yeah!
Oh, here we go.
There it is.
See the set? Here's the nucleus here, and there's the nucleolus. It's got a bunch of RNA nucleotides in it.
Oh, is that what subunits of ribosomes?
Yes, these will also make ribosomes.
That's a good
You
system.
make the ribosomes the same way. There's a portion of the DNA that these things add to, and then they will leave the nucleus and become a ribosome.
What?
But it's doesn't.
That's how the ribosomes are made.
By what?
By the same process I just showed you. We open up some of the DNA, we attach free nucleotides. The free nucleotides eventually will leave the nucleus and become the ribosome.
But doesn't it have to go through the endoplasmic reticulum?
It goes, not to make the ribosomes.
Okay.
Ribosomes are just simple.
Yeah, they're just pieces of RNA, and they're wrapped around some proteins. Also, I'm not showing you the whole thing.
Well, then what is it that the endoplasmic reticulum makes? I thought
What
it was.
is the endoplasmic
The endoplasmic reticulum is this big gloopy chamber that holds the proteins after they're made.
And the rough endoplasmic reticulum has all ribosomes all over it and smooth ones.
See all those yellow dots? Those are all ribosomes.
So
Okay.
It holds proteins that are made.
Yeah,
What does?
endoplasmic reticulum. It's a big storage area.
So this is where
Proteins.
this is where the this whole thing is occurring, is in the endoplasmic reticulum, because that's where the ribosomes are.
Yeah, but not always. Some ribosomes are just floating freely out in the cytoplasm.
Okay.
Some ribosomes are stuck on the edge of the nucleus.
There's also
But
polyribosome.
a lot of them are on the endoplasmic reticulum.
Because they're also polyribosome.
Yes, there are. Now wait, we have to do something else before we can leave the nucleus.
Take away the
We have to snip out introns. You remember introns?
Yeah.
Introns are useless bits of information that we don't need. They have to be snipped out.
Why are they there to begin with?
They were ones for like old traits that we don't need anymore.
That's right. They're evolutionary
And the tailbone.
Profiles?
leftovers that are not needed.
Wait.
Like you have like there's a lot of DNA for enzymes that are used in saltwater organisms. We don't need any of that information, so it ends up getting snipped out whenever we make.
And those are the exons.
And you cut out the introns.
You cut out the introns and the exons are left.
Oh, yeah,
Because they're going to be the ones that exit.
that's right.
Okay.
That'll help me remember.
They're expressed.
Yeah.
Sure.
Just
Yeah.
color.
I like Shelby's better, sorry.
I don't see how they help you anymore.
Okay, so it's hard to move around these things because they're not connected together. So I'll use this long string because that's easier to move.
Will you flip that one over? Thank you.
And that will leave the nucleus and we'll go to the ribosome. So we have finished transcription. Transcription was the making of this mRNA strand.
It isn't chopped, does it?
No.
Chomp.
Yeah, the ribosome?
Oh, chomp.
Because it looks like it should chomp.
No, it doesn't chomp. No, the restriction enzyme.
Now, again, like I said, the ribosomes are also made of RNA, and we call those rRNA. rRNA makes up the ribosomes. Also some proteins.
Wait, what is that space in there inside the black line? To the right, where it just came out of?
This hole,
Yeah, what is?
that's a nuclear pore. It's a hole in the nucleus that RNA can get out.
Can RNA just go to any ribosome
Yeah,
and it's the same thing?
yeah,
It just goes to the ones on
as far as I know.
That's the best option.
the
Now, there's one other type of RNA that's used to get the job done here. These little things that look like crosses, they're called tRNAs.
Tron.
They're also made of RNA. I know I have them green, but imagine they're a strand like this, all curled up.
And that's in the ribosome?
No,
Oh,
this is the ribosome.
okay.
These are tRNAs over here.
Question?
And they're carrying amino acids. They have these things attached to them.
Question.
Yes.
Is the ribosome thing, does it really have an opening like that?
Yes, as a matter of fact it does. It's actually like this at first. And then this thing will attach,
And then it
and then the lid kind of comes over.
Do you see what I'm saying there, Coach? That
It looks like teeth a little bit.
It seems like
That's just our model.
Like crunched down like a chomp.
Maybe, yeah. It wouldn't really look much like that in actual life. If you could
Did
see
you
something
make all
that
these
small.
magnets or did you buy it?
I made most of them.
I don't have any video on not a video on
Well, this is just I photocopied these and then had to cut them out.
from yesterday.
I just laminated them.
Then laminate, yeah, I laminated them. Cut them out. Okay, so what happens here? Well, this is translation. This is the second part. In translation, the mRNA, this strand, attaches to the ribosome and goes into the ribosome. And the ribosome has a couple of different sites here. The P side is on the left, and the A side is on the right. And I'll show you what those two things mean in just a little bit. But we have a name. There are 3 nitrogen bases here. 1, 2, 3. We call this a codon. 3 letters. And this one just happens to be, this codon happens to be AUG.
Is it always
AUG is what we call the start codon.
Is it always that?
It's always AUG. AUG is the first codon for every protein.
That's cool.
The start codon. And what's going to happen here is our tRNAs are going to come in one at a time into the ribosome and attempt to bind
Fail.
their bottom 3 bases with this codon. And we call those bottom 3 RNA bases on the tRNA, do you remember what we call them? The anticodon. Very good. So the way I would say this, if I were writing the essay, what would be, the anticodon of the tRNA attempts to bind with the codon of the mRNA.
If it fails,
The anticodon on the tRNA attempts to bind with the codon of the mRNA.
go to see one off it at first.
See ya.
Global.
Now, look, it's trying to bond, y'all.
But it can't.
It doesn't fit, does it?
Nope.
Nope.
No clue.
So it is rejected,
One blue.
and it leaves. So here comes another one. It tries to bond, also rejected. Here comes another one. It tries to bond, oh! Fits nicely.
Click. Click, click.
Click.
It's gotta click 3 times, good.
3 times?
Yeah, because
Oh.
Or I mean,
Click, click, click. Click, click, click.
or just one really loud click.
Click. Click!
There you go.
So, there it's set. And here's the first amino acid. Now, if this codon is AUG, what is the anticodon? Can you guess?
T. No.
What attaches to A?
T.
U.
Is it U still? I wasn't sure if it was because U.
What? Yeah, this is RNA also.
Okay.
UAC.
UAC.
Sounds like a university.
And the tRNA that has
Alaska
UAC, listen, Joey, the tRNA that has UAC can only carry one type of amino acid. It happens to be methionine. It's just something I know, you don't have to worry about.
So the UAC is always the first one to come in?
That's correct.
So methionine.
So methionine is always the first amino acid in the protein chain.
That's good for methionine.
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 How do you spell that?
METHIONINE. Methionine.
UAC?
Oh, UAC. Alright.
Now the next
Oh,
codon is exposed, and we just go through the same thing again. Another one comes in and tries to bind. Click. Oh, that happens to fit also.
Hey, that was convenient.
And when the 2 of them are next to one another inside the ribosome, a bond will form between the methionine
So cute.
and the next amino acid, whatever it may be.
So the
It's a
T.
pink one and a blue one, like a boy girl.
Just say peptide bond.
Perfect.
How many?
Now that bond actually takes an ATP to get it to bond.
Wait, isn't it like initiation or aren't there parts?
Yeah.
How many nucleotides would there be in a mRNA?
How many nucleotides in the mRNA?
Yeah.
It depends how long the protein is.
Got it.
The average protein might have 300 amino acids. that would be 900 nucleotides.
So there's a lot of tRNA.
Yeah.
Where's the RNA kept? Just like floating around in the cytoplasm?
Floating around in the cytoplasm. Now you'll see the same ones are used over and over though.
Yeah, sure.
What's that chain of amino acids called?
So,
Yeah, don't they go back somewhere? And do
once the peptide bond is formed, the first tRNA will exit the ribosome. It leaves its methionine because that's already bonded to something else. And now it goes back and it will get another methionine because that's the only thing it can carry. They're just floating around in the cytoplasm. Okay. They come from your diet. Whenever you eat, protein, your body breaks it down and it gets the amino acids get in your cells. So you can make more protein. You are what you eat.
Weird.
Okay, so now look, we're going to slide, the whole thing moves downward. The ribosome moves down a little bit.
Does the ribosome move or does the...
Well, I don't know. Either the ribosome slides down or the mRNA slides through. I think either can happen.
Or both?
It doesn't really matter. Both happen at the same time. And now the next codon is exposed. And something else may fit. Maybe this one. Oh, that one doesn't fit.
Oh.
See ya. Wouldn't want to be ya. Maybe this one fits.
Isn't it called elongation?
Click! That one does fit. Yeah, this is called elongation.
Another peptide bond.
Another peptide bond forms. The first tRNA leaves. And guess what it has to go get? Another what color?
Pink.
It has to get another pink. That's all it can carry.
Which amino acid is called?
The one I have is glutamine, but I don't know that that's correct. I just made a bunch of different colors, stuck them on there.
Okay.
So we move down again. And this will just continue. This happens over and over until we're done. Look what we're making.
Uh-oh.
Chain of amino acids.
What about the A and P site?
Okay, good question. I want you to notice we call this the P site. Why do we call it the P site?
Primary.
P stands for polypeptide, or you can say P stands for protein. There's a polypeptide, a protein, hanging from the P site.
There's a what?
There's a protein hanging from the P site.
That's cool.
See it right there?
Wait, what? Why is it called the P site again?
Because there's a protein, a polypeptide is also called a protein, there's the same thing, hanging from the P site. Do you see the protein hanging from the P site?
I see the protein.
You can't see it if you don't look up here.
The P
Wait,
site
is
is
that
like
just the
back
3
side of
amino
the
acids you're saying?
These 3 amino acids are the start of a protein.
It's
Is this the same thing as the P site?
This is the P site.
Okay.
The P site is a chamber inside the ribosome from which is hanging a protein. Do you see how the protein is hanging from the P site?
Yes.
Imagine this is the P site. There's a protein there hanging from it. Do you see?
Why is it? I don't understand that. Like, I get that it's hanging, and you said that like.
Okay, do you see that this is a protein?
Yes.
Okay, what if we did this for a long way?
Uh,
It would look like this.
Yeah.
Okay?
Isn't it like that? Like, through the whole thing? I don't understand.
Now, can you see that this is a protein?
Yeah.
Can you see that it's stuck here in the P site?
So it's not gonna keep going past that?
No. It's,
Nope.
They
It's
come,
always hanging from the P
they
site.
come to the A site.
The new ones come into the A site. The new amino acids come in at the A site. A site stands for amino acid. So the new amino acid comes into the A site.
I can deal with that. It's good.
Yeah.
This site is actually called the amino acyl site. Not amino acid
It
site,
sounds like you're
amino
saying
acyl site. And it
P
sounds like
site
the teacher when he's saying that is saying
asshole.
amino asshole. I remember all of us laughing at him. And he was in college and he was from a foreign country, so he didn't understand what we were laughing at.
Hey, Coach.
Because you're there spinning your
You would
book.
say another one comes to the amino asshole site, and he kept saying amino asshole,
Sound like
amino
Milton.
asshole.
It's like Milton from Office Space.
I don't know.
You don't know Milton?
Hey, Coach.
Oh yeah.
Yeah.
Stabler, burn down the building.
It's low battery.
Okay, hold on, we're low battery.
Wait, Mr. Willis, so
Yes.
just finish up real quick. So it moves down until all of it's done. Does anything happen at the very end of it, like the last week's next?
Yes, I hadn't got there yet.
Oh no, hurry up, I believe it. Here, you need to read this.
Okay.
There.
So, let's get to the end. Let's get to the end. Listen. Look at me. So this one will leave. We'll move down. Another one will come in. It doesn't fit, but just pretend. It attaches. The whole thing moves down. And eventually, we get to the last three. The last codon is called the stopcodon.
UGA?
And in this case, it's UGA. There are 3 stopcodons. One of them is UGA. There's 2 other ones. I don't remember what they are. But the stopcodon codes for...
It's
What?
A.
It doesn't matter.
I'm just telling you.
It codes for what we call a releasing factor, where this thing comes in and attaches. And it doesn't have an amino acid. And it'll cause this tRNA to leave.
And no more to come?
And nothing else has come. So when this tRNA leaves, you're left with your protein floating out here in the cytoplasm. It will coil up into its final structure, and voila,
And there you
you
have
made
it.
a protein. So that's how we make a protein. That's the essay. Look at all the ribosomes. Every one of these is making
See, I don't
a
like
couple
thinking about
proteins a minute.
That's cool.
that. That's too much, like, that's so complicated to think about everything that's going on at the same time.
You got it? Makey sensei?
Yep.
Okay, good luck.
Adios, Justin.
That's it?
Nice pink socks.
She doesn't wear.
That's it.
She doesn't know white socks.
Alright.
Questions about that one?
Let's move on to transcription.
I do.
We did transcription and translation. I'm done with both.
Whoa.
Wow. We're done early.
Transcription
Wow.
was copying the DNA. Translation was making the protein.
I have 50 minutes, sir.
We translated the code into a
So
protein.
respiration starts when the mRNA goes into the ribosome.
Respiration.
I mean, not respiration, translation.
Thank you, sir.
Translation starts when the
Yeah.
mRNA reaches the ribosome.
Okay.
Now another thing you may want to add, listen, you see this polyribosome?
Yeah. Yes.
That's many ribosomes in a line, and what will happen is a single piece of mRNA will go through all of them. And each one of them will make the same protein.
So it makes a bunch of proteins?
So you can make a bunch of the same protein.
That's cool. Oh, yeah, because
A lot of cells in your body are specialized just to make one type of protein over and over and over again. Like for instance,
What a boring life.
your stomach has a digestive enzyme that its cells create called pepsin. And it releases it into your stomach. So most of the cells in your stomach are just constantly making pepsin, one protein, and squirting it out in the middle of your stomach so you can digest your
I don't like that
food.
word.
So you have millions and millions of stomach cells, and they're all just making one protein, really. I mean, they make some others to help themselves live, but the one they keep excreting to the outside is pepsin.
But that in the polyribosome, it does the same, like translation, transcription, just makes the same one.
Yeah,
Okay.
yeah, all proteins are made this way.
Okay.
Okay. So the ribosome moves and not the mRNA?
They say the ribosome
It seems like the
moves.
mRNA should
It seems like the mRNA would move. I think both of them happen.
Alright.
I think both can happen. That's a good question. I've never really understood that.
And a particular just like strand of your DNA is just like it tells it ends up making proteins that do a certain thing.
Yeah.
Like,
Each protein has a job. Lots of them are enzymes that can build things or take things apart.
What's that?
So all you got to do is be able to make proteins, and then those guys can be your workers.
So you're like, hey, we need some carrier proteins.
That's pretty cool.
And then some
Then
RNA polymerase would go in there and find some
transcribe what we call a gene.
It finds a carrier
A
protein.
gene for making carrier proteins.
Nice.
Yeah.
So it's kind of like going to the library and finding a book and then
Exactly.
read that.
Exactly. That's the DNA is like a library that has a gene is like an instruction manual for making a protein. There are 20,000 different genes, which means 20,000 different proteins can be made. And they're all running around inside your body doing their little jobs.
That's why I don't like to think about it.
And that keeps you together.
And say that could be just like the gene that says you have brown hair that just happens to also make a carrier protein thing. Or something like that.
No. In actuality, this is really, really long. And you have this section is for the brown hair protein, and this section is for a carrier protein, and this section is for a different protein.
You said the brown hair protein.
Because
It's called melanin.
It was in your skin.
It makes your skin dark, it makes your hair dark.
Wait, so but there's different ones for each, obviously.
the proteins are actually, they're like the thugs.
Yeah, I don't know.
They
It
go
gets
do the
it gets
thing, and you get
complicated.
it done. The DNA is like, you know, the mastermind.
Nice.
The DNA is Don Corleone.
I'm writing that in like my national essay if I get this.
Okay.
Compared to the mom.
Coach, am I on guys on target here?
Huh?
I said the RNA is like the thugs that like go like do what needs to be done, but the DNA is like, you know, the mastermind or whatever, just sitting in the back telling people what to do.
RNA is like a messenger. So if Don Corleone said,
Go tell.
go collect for me, or send a message to have that guy killed,
And
I
the
was gonna
proteins
say-
are the thugs.
then the RNA would leave and take the message.
Protein are the thugs.
The proteins are the actual workers.
Yeah,
So the proteins
guys
are the
like
ones who
bam.
like, yeah, they're the
The proteins do the work. They're the enzymes.
Proteins
So they're the
do
people that you
guns
hire
and
to
take the
kill
cannolis.
in this scenario.
Let's not write about it like that. If you get this. Okay, that's it, I guess. Any questions about anything? Can you handle
How
that?
was your day, man, dude?
Will this out?
Pretty good.
Alright.